We are here, that is, we exist, possibly because of phage. So rather than speculate on the future 50 or 100 years of phage biology, I'm going to do something that I'll consider maybe more conservative. I'm going to tell you what happened a billion years ago. Okay. And to tell this story, I need to introduce the phage that we've been working on for a while. And to do that, I'm just going, let's see here, to do that, I'm going to figure out how to work this. And I'm not going to figure out how to work this. Oh, there we go. To do that, I'm just going to sort of remind everyone that we all know that phage have been intensively studied. Thousands, maybe tens of thousands of phage genomes have been sequenced. But the vast majority of the phage that have been characterized actually have relatively small genomes. I went to a website, pulled up 1,604 phage genomes that were in the website, made a histogram of genome size versus frequency, and the vast majority of those phage are under 200 kb in size. The average is somewhere around 50 kb. But there are a lot of phage out there that are much bigger than this, and those are the phage that caught our attention a while ago. These phage go from 200 to 300, even 500 kb. So they're approaching the size of a minimal bacterial genome. And we got interested in these phage because we noticed that many of them encode a homolog of the cytoskeletal protein tubulin. So we started working on them, and to put these tubulins into perspective, I'm going to show you a phylogenetic tree that has some of the main families of tubulins represented. Up here on the left, we have eukaryotic alpha-beta tubulin. They, of course, make up microtubules that assemble the spindle that's essential in all eukaryotic cells. Down here, we have FTSZ involved in cell division. We've got TUGZ over here, which is a tubulin involved in plasmid DNA segregation. And then we have a variety of other really divergent tubulins. The ones in green up here are all encoded by phage, large phage, that infect strains of Pseudomonas. And so we thought, well, this is really curious. So we started working on them. And the first one we started to work on was GP59. It's encoded by a strain that we just refer to as 201. It has a longer name, but that's a little bit easier to say. It's 316 kb in size, and it infects a strain of Pseudomonas called Chlororhynchus. And so we started working on this phage and on the tubulin encoded by this phage, and we set out just to answer the question, why would these large phage all be carrying a tubulin homolog? What possible function could this tubulin be playing? And we didn't know at the time. But we now know, we think we have the answer to that question. And I'm just going to summarize what we found. We've done a lot of experiments in my lab using cell biology and genetics, and we've collaborated with a structural biology lab at UCSF, the lab of Dave Agard, where they've purified the protein, characterized its polymerization dynamics in vitro, and solved the structure of the protein and the filaments that it assembles into. And so just to tell you a little bit about what these tubulins do, if you take and you make a GFP fusion to these proteins, which we call fuzz, and you express it in the cell, during lytic growth, fuzz assembles into dynamic polymers. And so what we have here are 5 examples. The dotted lines here sort of outline individual Pseudomonas chlororhynchus cell that's been infected. Each cell has a pair of filaments, and it seems like the filaments originate from the pole of the cell and move towards the middle. Over here we have one cell that's been enlarged. It's a 3D reconstruction obtained with the OMX structured illumination microscope. And what you can see is that you have a pair of filaments that sort of meet at a vertex. This is the one pole of the cell, this is the other pole of the cell. And those filaments appear to reach towards the middle, suggesting that during phage infection, you get some kind of a structure like this assembled, where you have basically a bipolar array of filaments reaching towards the middle, and that sort of immediately makes you ask the question, well, what's in the middle of these cells? It turns out that in the middle of these cells during phage infection, if you stain the cells with DAPI and look at the DNA, there's a massive ball of DNA. That's all phage DNA. The host DNA is degraded. We know that from fluorescence in situ hybridization experiments. And on either side of this blob of DNA, which we have termed the infection nucleoid, you have these polymers of the fuzz spindle. We believe that these fuzz polymers are playing a direct role in positioning the infection nucleoid, because if you make a mutant that disrupts the polymerization dynamics of the protein, so it can assemble polymers but they can't disassemble, it severely mispositions the infection nucleoid. And in this condition, individual burst size decreases by about 50%. So that allowed us to propose a model for how these particular phage replicate, where phage DNA enter the cell, we believe at random positions. We can watch phage DNA after it enters the cell migrate towards the middle of the cell. We believe that migration is being driven by the polymers that are set up of fuzz by the fuzz spindle. The phage nucleoid grows and increases, replicates, and increases in size in the middle of the cell, and that's where we have evidence that capsids assemble, DNA gets packaged, and so you end up with basically a sphere of viral particles assembled in the center of the cell, and ultimately the cell lyses. And so once we found this, and we had this spindle-like structure assembled in the cell, we said, well, you know, there are no other such spindles that have ever been found in a prokaryote. In fact, the closest thing that this looks like is a eukaryotic spindle. So I'm going to put that image of the eukaryotic spindle up there and say, is the fuzz spindle produced by this phage, is it actually really biochemically similar to a eukaryotic spindle, or is this just sort of, a superficial resemblance? So here's that image again. Every eukaryotic cell, without exception, has a bipolar spindle made up of these microtubules, and they come in a variety of shapes and sizes depending upon the cell types, but they all have the same key properties. First, you've got an array of filaments that are made up of tubulin, and these filaments have distinct structural and biochemical polarity. There's a minus end to these filaments, and there's a plus end. The minus ends to these filaments are usually anchored in one specific location in the cell, such as a spindle pull body, and the plus ends are really the business end of the microtubule. The plus ends undergo cycles of growth and shrinkage. So they'll grow for a while and sort of randomly switch to shrinkage. And this ability to grow and shrink, called dynamic instability, allows a microtubule to search the space of the cell, and if it happens to bump into and connect with a chromosome, such as a kinetochore, it can take that chromosome and position it in the middle of the cell. So these key properties, it turns out, we now know after 5 years of work, the fuzz spindle has all of these properties as well, making it the first example that we know of a bipolar spindle that's set up in a prokaryotic cell that has all of these features. And I'm just going to tell you a little bit about the spindle structure. So first of all, Dave Agard's group solved the structure of the fuzz monomer. That was James Kramer, a graduate student. And then another graduate student, Elena, solved the structure of the entire filament. And they found that the fuzz monomer has the typical tubulin fold that binds GTP, but then it also has an extended C-terminal tail. And in collaboration with us, we were able to show that this C-terminal tail actually plays a key role in fuzz assembling a unique triple-stranded filament, first triple-stranded tubulin filament that's ever been found, where protofilaments sort of gently wind around each other. And just like a microtubule has a plus and a minus end, these filaments also have a plus and a minus end, where, in fact, where the plus end displays dynamic instability inside the cell during phage infection. The minus end, we believe, is anchored at the cell pole at that vertex, and that the plus end then is oriented toward the middle of the cell, displaying dynamic instability where it can grow and shrink, and thereby position the DNA of the infection nucleoid in the cell center. Why would the phage want to position the DNA in the center to begin with? Well, we believe, we don't know for sure, but we believe that this contributes to viral replication, and that maybe once you reach a certain genome size, and so if you have a larger genome, you have a larger capsid, that you actually, just diffusion alone makes it a less efficient process in order for the DNA to get packaged, and so you can probably much more efficiently produce phage in factory-style replication. Okay, to put this triple-stranded filament sort of into perspective what we know about other tubulin structures in bacteria, over here, oops, we have FTSC, which forms just single protofilaments. TUBZ, involved in plasmid segregation, has been shown to form double-stranded filaments, and then more recently, four-stranded filaments. And we believe that these, it's kind of easy to imagine that these triple and four-stranded filament structures could be evolutionary intermediates on the way to building a eukaryotic-style microtubule, with 13-stranded filaments. And what's interesting about this is that the thicker you are as a filament, the more force you can generate without buckling. So these bigger filaments are going to be able to push a much larger payload, and therefore a much bigger chromosome. And so now this is where the title of the talk comes into play, and where maybe I'll really go off the deep end on speculation. And I'm going to talk a little bit about why we think this phage spindle is so interesting. The eukaryotic spindle is a really complicated structure, and every cell has one. So it's thought to be a structure that arose very early in the very first eukaryotic cells, and it provided a structure that was strong enough, that it could provide large enough pushing forces, that eukaryotic cells could have much, much larger chromosomes than prokaryotes have. But where did it first come from? How could it possibly have just suddenly evolved to have all of these amazing properties and assemble and start pushing chromosomes around? And we've never found one in a bacteria. The fact that we found one in a phage allows us to propose for the first time the first plausible explanation for how the spindle might have arisen, where you might have had FTSZ and tubulins evolving early on for the purpose of cell division, making really rudimentary filaments. These could have evolved into higher order filaments, such as these double-stranded filaments involved in plasmid segregation. Ultimately, we ended up with triple-stranded filaments like Phage that have all of the key biochemical properties and actually perform a similar function of taking DNA and positioning it in the center of the cell. And once you have a structure like this, which serves a specific purpose for the phage, organizing viral replication, it's not much of a stretch to imagine that maybe at some point, instead of grabbing and positioning phage DNA at mid-cell, that it might have grabbed and positioned DNA in a very early cell, a bacterial cell, or whatever that progenitor cell you want to call. And perhaps when it did that, if it did that, that would even serve some selective advantage, such as keeping chromosomes closer together, which could maybe stimulate recombination, which maybe would, for example, provide increased resistance to UV. So this is sort of a hypothetical intermediate that could have gone on to produce, evolve into what we know of as the eukaryotic spindle today. So over time, we have an increasing degree of filament complexity, biochemical complexity, and structural organization. Now, I'm proposing that phage, since they're the only structures in bacteria that we know of that are certainly related to the eukaryotic spindle, and they form a spindle, that the phage spindle might have been what has evolved, gave rise to the eukaryotic spindle. But of course, there's alternatives. Maybe, we all know that phage are capable of grabbing other stuff from the chromosome. Maybe the phage stole this from the spindle-like structure from some bacteria. That's a possibility, but we've never found one in a bacteria, and we certainly believe that bacteria don't rely upon at least tubulin-like structures for chromosome segregation that do this kind of spindle organization. Of course, it's also possible that maybe the phage stole the spindle-like structure from a eukaryotic cell, but then again, that still asks the question, where did the eukaryotic cell get it from? Since phage and bacteria predate eukaryotes, I think it's simpler to imagine that phage might have evolved at first for one purpose that could have then given rise to the eukaryotic spindle. And I guess while I'm on this slide, I want to make maybe one more point, and that is in the 50s and 60s and 70s, phage played a really important role in as just tools in establishing the central dogma of molecular biology. Today, phage and plasmids are also playing central players in establishing the central dogma of prokaryotic cell biology, just helping us define what is the prokaryotic cytoskeleton and what does it do. And so, if I had time, which I don't, I was going to tell you kind of what we're doing next. We're looking for other factors involved, and I'll just summarize. We're finding other proteins that are produced by the phage that we expect might participate in formation of this replication factory in the center of the cell. And in fact, we have some evidence to suggest that the Phage spindle might be involved not just in positioning the DNA in the center of the cell, but also in helping the capsids, which assemble in the cytoplasm, find their way to the infection nucleoid. And so I'm just going to stop here. This project was started by Marcy Erb, a wonderful grad student, and it's being followed up on Vorapon, who's in the audience there, as well as Katrina Nguyen. And so I'll take any questions.